Yeah. Okay, so we can start. Tomáš. Tomáš is back, so questions please. So, okay, so Roel was first.
So my main question. Oh, I have to talk. I don't have to shout. Okay, yeah, perfect. Okay. So you mentioned that the chicken was taken as an example for all avian and non-avian reptiles, and I wondered in this the phylogeny you covered now of amniotes, including your future plans, didn't include any birds. Is that because they are very well studied already, or is it really true that the chicken is modeled for all avian species?
I have also a collaborator for birds, but that's not happening in the next 2 years. But I will study birds as well. We do know that quite a few bird species look like chicken, but yeah, it hasn't. I mean, I would be very interested in further distant species. And even if they look like that, the interesting thing would still be the gene regulatory networks for me. Whether the gene regulatory networks are the same between the different species or not. So I'm definitely going to go for some birds as well.
Right. I will switch it to here.
Okay, thank you very much for a very inspirational talk. I would like to ask you, because you are mostly interested about the pre or the pre-gastrulation period, but also reptiles differ because they don't have primitive streak, unlike birds. So is it possible that the pre-gastrulation development could be somehow also like related with not having the primitive streak, or does the different things you are seeing in, for example, chameleons?
I would say it is not because they don't have a primitive streak, because we have such a diversity in mammals, and they all have a primitive streak. So if it was something that was streak or non-streak dependent, then all mammals should look very similar, and they don't. So, yeah.
Yeah, my question was more about birds and non-avian reptiles, because they then gastrulate differently by more like involution, and if it could be somehow that it could be related or not.
I don't know. I don't know yet.
Oh. Okay. Thank you.
I think I need more species with different shapes, and I need them towards a point where I can actually do some tissue mechanics studies, which the staging series is the first step to then be able to define the stage of interest, to then start studying that in more detail, and I hope that eventually I would be able to answer that.
Maybe it's completely out of topic, it's just a curiosity, because like with the people that I was working with, they were saying that I don't know, maybe it's not something that you study, but that like in mammals there is some also inheritation of the microbiomes. So I just came up to my mind if you saw something different with the oviparous regarding the transmission, or if it's not something that you can see with this type of analysis. I don't know, I don't know how they work, so.
So, double check, you're asking for whether there's an impact of the microbiome?
Yeah, no, like if, because with mammals, they study a lot how they transmit it, how the mom transmits it to the embryo. And I was wondering if it's something that they also look in oviparous, and if it's something that you see with this type of techniques, or if it's something outside of...
It wouldn't be visible with these techniques. It will be visible in the moment you do sequencing, whether you could find genomes that wouldn't be that would not be from the species. I'm not focusing on it right now, but I think it would be very interesting to understand how 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 anything that's in an egg would get their microbiome, and whether there's any sort of transfer between mother and the neonate for sure. But yeah, it's a bit beyond what I'm doing right now.
So I have a comment to it. My friend Natalia Feiner discovered a helminth that is it's living in the brain of the mother, then it colonizes gonads, and it goes to the brain of the embryo, and it's being transmitted in oviparous lacerted lizard like this. So they publish it in American Naturalist, and they say they don't want to work on it further. But it's even like big animals, like helminths, can go through oviparous, like can go vertically in oviparous lizards, which is fascinating to me. But I have somewhat related question. So why is such diversity? So does it not matter too much, like what's going on before gastrulation? So it's something, some movement are fine, and then like one would say that such important process would be pretty conserved, but you show just in mammals, and like you touched two more or less randomly chosen reptiles, and it was totally different. So why this diversity? What do you think about it?
I don't know, because the interesting thing is, if something does go wrong during that time period, so if that random shape is not achieved, then the embryo aborts. So yes, we have the different. I think it's coming a bit from the hourglass of evolution. So I think that the shapes in the beginning are massive. We go towards the similar shape during neuronation, and then we have the final phenotypic stages. I don't know how they developed, but I presume that. I'm very interested to see whether there are any sort of signaling differences between the chameleon or any other species that is doing a lumen versus other species, because that would, and whether this is in any way advantageous, or whether something else is playing a role. Chameleons do very, very slow embryo development. The anole is quite fast. Maybe the chameleons have more time to make such a sophisticated shape, but at the same time mice do it within a week. So that would be even faster. So I think it's very tricky. I think it's just
like
something must have optimized. These species must have optimized for something in this direction. And I think once we have different shapes, we have this a bit more established. Doing perturbations of one species and trying to force one species into the shape of the other, and see what happens then. I think that would be very, very interesting.
Something with cell size, that migration is easier somewhere. Or
cell cycle length, or something.
Maybe can be somehow related also to like maternal investment or so, or development style, or like if they have many eggs or a few eggs, or I don't know if that's any pattern you observed.
Not yet, because I have not sampled enough species. I think most large clutches are not that common for reptiles. I mean, most from the turtle collaborators I have, most turtles would be between 8 and 15 embryos, which also wouldn't go towards the 50 to 90 of a chameleon. So I don't know.
So just as speculation, I would say that mammals can more like play with that, because they can filter out all the bad embryos. As you showed, 60% of embryos go out, and the investment is like nothing. However, in reptiles, all the yolk is already there, so they should be more careful, I would say, because the loss is enormous. Even a single egg is quite big, like investment.
And that 60% is true for humans. Mice don't have that low. They have a much higher success rate of embryo development. But yeah, maybe.
Thank you for the lecture. I have one comment or maybe answer, a suggestion of answer for this huge variability, not just in reptiles, but in the whole phylogenetic tree. I think a lot of this can be some kind of vestiges of various successful or sometimes unsuccessful genetic takeovers events. Some population of embryonic cells try to start to be germline, and therefore it results in such huge variability.
Seems to be clonal, but still.
Thank you very much for a wonderful talk. I will have a question concerning the molecular markers you used for the whale chameleon. Was it whale chameleon, right? So you studied, or you took a look at the nodal pathway, but I was just wondering, there is a genome published, right? And you show only one nodal and one lefty, so are you sure that there's only one paralog of each of these? Because the lefty, yes. there may be different roles of the different lefties, for example in mouse.
The lefty probe that I used was covering both lefties, and the nodal 2 I showed, the nodal 1 I also did that. It looks quite similar. So there are two nodals in chameleons, and they both they don't have the perfectly same pattern, but they're quite similar. And from the lefties, none of the lefties nothing was on the same for the Cerberus. Mm-hmm. The probes I was using, they were covering both.
Okay.
Both genes.
So did you check also other members of the pathway, like GDFs and DAN-D5, if
no,
there's one?
No, because we felt, so even though we do get 50 to 90 embryos per clutch, it is actually not that many if you want to do this kind of study with it. So we are currently waiting for single-cell sequencing data set to then, because right now we were going for the canonical ones, and since it's not them, we can now continue picking out, or we just wait to see what it is. Because I felt by now we will just waste a lot of embryos to look for.
Yeah. Did you also check SMATs? Because all these markers are just, extracellular players, but the SMATs could actually tell you the real activity of the pathway and the target tissue.
SMATs are tricky to stain. So these were all in situ hybridizations, which you designed for the genome. The antibodies are usually raised against mouse or human or something.
But I know that Natasha Shiloh used SMATs for her left-right pathway studies.
We did not, the probes there we did not do it because for the same reason, we did not have that many embryos in the end. Because we also were doing quite a few IF stains as well that it didn't show. So we decided to rather wait for the single cell seq.
Yeah, and one more question regarding the brachyury and the circular and then more focused signal or expression pattern. It reminds me a little bit of fish stuff, where you have, during gastrulation, you have this epiboly, and during that epiboly you have a circular expression of brachyury, and then it restricts to the shield stage, and then to the forming notochord. Yeah. And that's exactly what reminded me, or what your data reminded me about.
No, exactly. But the thing is that we have brachyury expression before gastrulation happens, because gastrulation is for that we actually need first an epigast that is established. And we need the breakage of the basing membrane. We need all of these points that have not been ticked before. What I think is happening is that during that lumen closure, the cells that start ingressing and closing, that they undergo somewhat of an EMT in order to close the lumen, and thereby then starting to express the mesodermal marker, which is brachyury. But that we have a second role of brachyury, or just as a marker expression, we have also now staining of brachyury that is, so protein staining of brachyury, so that really is there. That it is more a marker of the identity of these cells, and that that we have a two-step process. We have one brachyury upregulated for EMT to close this lumen, and then we have anterior-posterior patterning and brachyury upregulated for gustulation. Yeah, okay. But I agree, it was from the ring, it looked quite similar to fish, but again, amniotes are not really supposed to look like fish. So.
Sure.
Yeah.
Thank you.
Thank you. Okay.
Which is about, so now you showed quite diverse phylogenetic, well, at least from different groups, but do you have any insights or ideas if, for example, in all anoles it's conserved, or in all chameleons it's conserved, or in all, let's say, primates it's reserved? Or?
So, my current plan is to have, to study in depth one species per family, and to then take another species and check. And the first time we can actually do it properly is with the geckos, because we have the morning geckos and the Paradoculus. So we have, for the first time, 2 species of the same family. I hope that it would be conserved within one family, but you never know. We'll see. But I would hope that I could presume that the families would be fairly similar. For the mammals, I will have quite a few with all of the deer. They will all be quite close, but not for the others yet.
Okay. So, it seems it was the last question. So, thank you once more.